can't get them to stay close together. So this is what keeps the lipid droplets emulsified, suspended, as small components. And we'll do this in lab today with vegetable oil and biosalt. So now the enzyme that interacts with the lipid is going to be pancreatic lipase. Again, this is the enzyme we're using in lab today. So it needs to be a alkaline pH. So within the lumen, pancreatic lipase and water interact with the micelles, and these are broken down into monoglycerol and fatty acids. All right, so those are our components. These are still fat soluble. And so they move into, they don't need special transport proteins because they're soluble amongst the phospholipids of our cell. And so we have our monoglycerol molecules and our fatty acid molecules now inside the cell. You would think we would be done with this now, all right? No. These resynthesize. So these come back and form triglycerides again. So those are resynthesized in the smooth endoplasmic reticulum. And they pass into the Golgi, where they become packaged with the plasma membrane. So now we have our triglycerides. Remember how if we make proteins and they go through the Golgi, they get pinched off, surrounded by a membrane sac that directs them to the mitochondria. If they're enzymes for the mitochondria, directs them to the surface of the cell if they're digestive enzymes or antibodies. So now instead of being directed to the surface of the cell, these are directed to the basal membrane of the cell, where they fuse. And via exocytosis, because again, they're still, I left off one step. In the Golgi, sorry about that, in the Golgi, they are covered with a protein coat. HDL lipoproteins, HDL, VLDL, LDL. Why do we need to put a protein coat around this lipid? Are proteins polar or non-polar? They're polar. So as a lipid droplet, this is not going to be soluble in the fluid of blood. With a protein coat that is polar, that can interact with the water and increases their solubility. Otherwise, we'd have lipid embolisms, little fat droplets traveling through the blood. So now our protein-coated triglyceride is exocytosed because it's way too large now to pass through the plasma membrane. And this is known as a chylomicron. The protein-coated lipid is a chylomicron. And as a chylomicron, it is way too large to diffuse into the plasma membrane. So guess where it goes? What vessel is large enough to take this protein because it's large enough to take whole cells? The lacteal. Our lymphatic capillaries, remember, have the cells that will create a gap. And so the lipid, protein-coated lipid, goes into the lacteal. So our absorbed amino acids and our absorbed simple sugars are taken up into the capillaries of our villi, whereas the absorbed lipids with their protein coat are taken up into the lacteal. All right, so back to this histology model. Here's the lacteal. So lipids are going into the lacteal and into the lymphatics, and then the lymphatics are going to carry it where? Right side or left side? Right side or left side of the body? Pardon? Left side. So it goes into the cisterna chyli and up the thoracic duct, into the subclavian vein, to the heart, pumped out the aorta. And so how does it get to the liver? The liver needs it for storage of fat-soluble vitamins or gluconeogenesis. Hepatic artery. So our hepatic artery is rich in lipids and oxygen, whereas the capillaries that have been picking up the amino acids and the simple sugars are going to form the superior mesenteric vein, which is going to flow into the portal vein, and that's how it gets into the liver. So our portal vein is rich in amino acids and simple sugars. Our hepatic artery is rich in oxygen and lipids, and they're both flowing into the liver. So that's why we need both of those vessels to carry to the liver, what was carried to the liver in fetal life by the umbilical vein. So we just needed that one vessel in fetal life. We need both of them, portal vein and hepatic artery, after you're born. All right, now some forms of, and I'll see if I have time tomorrow or maybe on Tuesday, because I did talk about it with the morning class. You guys are aware of gastric bypass surgery, okay, or lap bands. With a lap band, the stomach is kept intact. There are actually more severe forms where the duodenum is cut out completely and the ileum is attached up to the stomach. You completely remove the jejunum and attach the ileum to the stomach. Now that severe form, we miss. You look at it, it's like, this isn't going to work because you're missing the input of bile and you're missing the input of the pancreatic enzymes. So some other forms of surgery will, I need a stomach diagram. They will take, and I'll put some of these diagrams, show them to you either in lecture tomorrow or Tuesday. But they'll take the stomach, and then they'll take, here's our large intestine, they'll take cut out the duodenum, and bring the ileum up here, and then they'll take the duodenum, cut that off here, but and clamp off, so they'll have one end of the duodenum clamped off because this was put in its place. But remember, the duodenum receives the liver, bile salts, and the pancreatic juices, and they'll bring that part of the jejunum way down here. So you do get the bile salts and the digestive enzymes, but so close to where the ileum is going into the large intestine that you have a very short time for digestion and absorption to occur.
Passing it all back out.
Yeah, and you just excrete it. So there's a variety, excuse me, of these different types of surgeries, and you wonder how anybody could have a normal.
You definitely want to chew well.
Yes. been somebody who's prepared for a week for colonoscopy. It's not usually that clean. So one of those salt flushes, so it came out clear.
You know, not to change the subject, but yesterday I think it was, I was watching a show where they were doing coffee.
Yeah.
Why, I don't know, but maybe the diuretic factor of the coffee or something?
Yeah, or the effect on blood vessels. I tried that salt flush this weekend and all I did was gain 2 pounds of water. Which according to what I was reading, I was like, if I'm going to talk about this stuff, I should know what I'm doing. It was just like, okay. I think what I did wrong though, was you were supposed to put, the recipe I had was 3 teaspoons to a tablespoon in 2 quarts. I did it in 1 quart. So that might have been why. But anyway, I decided that was enough experience of that. So large intestine, notice the pH is still alkaline, all right? So we have the absorption of food products of finished. We don't have we don't have villi. We don't have a lot of the microvilli. We have way more goblet cells. And I was reading the other day, one of the reasons why it's 25%, but it looks like more, is the goblet cells are so fat that they squeeze the other cells, and they look thinner, so that it doesn't look why they're quite as many as they are. So we do have an intestinal flora, E. coli, that's going to be using, it's a symbiotic relationship. Their enzymes will break down some of the non-soluble fibers that we have and the plant fibers, and they produce a vitamin K. And that's about it. We're getting vitamin K and water from our large intestine, okay? Unless you're constipated and impacted, and then you start absorbing bacterial waste products into the body, which is not a good thing. And we talked just a wee bit about defecation reflex on Tuesday. This diagram goes into it in a little bit more detail. I'm just going to write the nerves that are involved on the board. You guys thought we were all done with pelvic splanchnic nerves? So you've been reading up on the micturition reflex for the take-home exam, and the stimulus for the rectum is the same as it is for the bladder, stretch receptors, right? So as fecal matter moves into the rectum, we have stretch receptors in the muscular wall of the rectum, and this is going to excite sensory neurons. Many of these will be carried along the pudendal nerve. Via inferior rectal branches. But some will be carried along with sympathetic and parasympathetic fibers. Remember, sympathetic and parasympathetic fibers are always and only motor neurons. So we're not saying that they're sensory neurons, but traveling with those motor neurons, just like we can have sensory fibers in the pudendal nerve travel with lower motor neurons, we can have these sensory fibers, all right? These move to the spinal cord, where they carry the impulses to the spinal cord, and this would be the sacral spinal cord. Where they synapse with and it should be a 2-neuron system, so there would be another neuron in the wall. And these would be parasympathetic fibers. So parasympathetic fibers are the pelvic splanchnic. And they are going to be excitatory. So their effect of releasing acetylcholine is going to cause an increase in the action of our circular and longitudinal muscles. And we're going to see an increase in waves of peristalsis passing through, moving the fecal matter down the rectum. At the same time, we have additional pelvic splanchnics that are coming to the internal anal sphincter. Remember, that is smooth muscle. And now the effect of acetylcholine is going to be inhibitory. And the internal anal sphincter will relax. So all of this is overriding the sympathetic effect, which would be the opposite effect. However, you may, wake up in the morning and you know that you need to go to the bathroom and have a bowel movement, but then, the dog starts chasing a robber outside your window, or the faucet breaks, or your kid, has stopped at the toilet and you have to go unstop it. And so you don't respond to that first urge. All right? So what's keeping the fecal matter from leaving your body is the fact that your external anal sphincter is still contracted. That's a voluntary motor, it's a skeletal muscle. And so what is activating that is inferior lower motor neurons. Going to the external anal sphincter. Have to be inhibited. But the inhibition occurs up here. We inhibit the lower motor neuron from firing. Because when it fires, it's always going to be active. It's always going to cause the skeletal muscle to contract. So here we have the acetylcholine release is binding to smooth muscle receptors in the smooth muscle and causing them to relax, just like acetylcholine causes cardiac muscle to become less charged, to lose its positive charge. Whereas here, we don't do that at skeletal muscle. Acetylcholine is released, it's always excitatory. So to inhibit that, we have to inhibit it up here. We have to have some other neuron inhibiting the lower motor neuron. And that's where we have a cerebral effect. Okay? So thought processes, being busy, going, no, this is not the time, I'm driving the car, that's not going to work, or whatever, you have to stop that. So we're constantly sending positive excitatory influences to our external anal sphincter until it's an appropriate place or time, and then we inhibit that, and then the external anal sphincter will also relax. So that's kind of a brief summary. It's very similar to what we've described before, using those both autonomic and voluntary motor fibers. Okay, so that's the end of lecture.